Today, we're diving into a kind of scientific detective story. We're going to crack a case that's flipping a fundamental rule of neuroscience on its head, uncovering a secret life in brain cells we thought we had all figured out.
So let's meet our main character, the oligodendrocyte, or, OL for short. Now think of these cells as the brain's master electricians. Their big job is to wrap up our nerve fibers, all the brain's wiring, in this fatty stuff called myelin. And this myelin sheath, it's just like the rubber insulation on an electrical cord. It lets signals zip through the brain at unbelievable speeds.
For the longest time, science basically saw these cells as just quiet background support. Super important, yeah, but passive. They built the insulation and, well, that was pretty much their whole story. They were considered non-excitable, which is just a way of saying they couldn't generate their own electrical signals, not like neurons do.
But what if that whole story was wrong? What if these quiet little support cells have a secret life? One where they aren't just passive insulators, but are actually active players in the brain's electrical chatter? What if they can spark? And that one question, well, that's what leads us to our first major clue.
Okay, so our investigation kicks off with the very first clue, a totally unexpected spark of life found right where nobody even thought to look.
So get this. Researchers were studying a part of the brainstem that's absolutely jam-packed with these insulated nerve fibers, and they stumbled onto something that should have been impossible. A special group of young, still developing oligodendrocytes, they're called pre-OLs, were doing something they had no business doing. They were generating electrical spikes, just like a neuron.
And we're not talking about some weak little flicker here. The study actually calls it a bona fide action potential. Now, that's science speak for this is the real deal. These were powerful, all-or-nothing electrical blasts that looked almost identical to the signals that neurons use to talk to each other. This was the first piece of hard evidence that something completely new was happening.
Now, to really get how big of a deal this is, just look at this chart. It's comparing these new sparks to the weak little electrical blips we already knew about in even younger cells, the OPCs. See that top row, spike type? The old precursor cells just have a weak little fizzle. But these new POLs, they've got an all-or-none action potential. That's a true blast of energy. The spike's peak is way higher, and the sodium current, which is like the fuel for the spark, is so much stronger. This isn't just a small improvement. It's a totally different ballgame.
All right, so we've got a spark. Now, in any good detective story, once you find a clue, you got to figure out how it happened. What's actually lighting the fuse? So our investigation now zooms in on the tiny machinery behind this amazing ability. So the investigation pretty quickly zeroed in on a prime suspect, a very specific protein called a voltage-gated sodium channel. These things are like tiny electricity-sensitive gates on the cell's surface, and the one specific channel responsible for this spark, it was identified as NaV1.2.
And the way they proved NaV1.2 was the culprit? Oh, it was just beautiful science. They used this highly specific toxin that blocks only NaV1.2 channels and nothing else. The moment they put it on the cells, the electrical spark just vanished. Gone. And to really seal the deal, they stained the cells and could literally see the NaV1.2 protein right there on the surface. That, my friends, is the smoking gun.
Okay, so we found the machine that makes the spark, the NaV1.2 channel. But that just opens up the next big mystery. Are these cells just sparking randomly, or is something else lighting that fuse?
And this is a really critical question, right? Is this just some random biological noise, or is it actually part of a bigger conversation? The next phase of the investigation was all about finding out if these glial cells were actually listening to signals from their neighbors, the neurons.
And it turns out they are 100% listening. This little flowchart lays it out perfectly. It all starts when a nearby neuron gets active and releases a chemical messenger called glutamate. Step 2, that glutamate latches onto special receptors on the oligodendrocyte. Step 3, this causes a little electrical change. Now here's the crucial part. If that little change is big enough to hit a tipping point, Step 4, it triggers those NaV1.2 channels to fly open. And bam! Step 5, the oligodendrocyte fires its very own action potential. This is a direct conversation from neuron to glia.
And this isn't just a random thing either. When the researchers looked at when these sparking cells show up during brain development, they found this amazing pattern. The number of these excitable cells just skyrockets at exactly the most critical time for building myelin. And they don't just vanish, a bunch of them stick around into early adulthood, which really suggests they have an important ongoing job to do. So, this brings us to the grand finale of our story. We found the spark,
We know what machine creates it, and we know the neuron signal that sets it off. But the biggest question of all is still hanging there. Why? What's the point of all this? To solve this final piece of the puzzle, the researchers came up with one last big experiment. They asked a really simple, direct question. What happens if we just turn the spark off? If it's really as important as it seems, then silencing it should have some kind of real, measurable consequence.
So what they do? They use this really clever genetic tool to go in and specifically shut down the NaV1.2 channels, the source of the spark, but only in these developing oligodendrocytes. Basically, they cut the wire to the spark plug, making these cells electrically silent.
And the results were just, wow. They were visually stunning. On the left, you see a healthy, normal cell. Look at that complex tree-like shape reaching out to connect with things. But on the right, look at this silenced cell. Its growth is totally stunted. It's smaller, with way fewer branches, and the ones it has are shorter. I mean, it's crystal clear. Without the ability to spark, the cell just can't grow up properly. And that physical stunting had a huge functional consequence. This bar chart tells the whole story. It's showing how much myelin protein gets made. The tall bar on the left, that's the normal, healthy group. The bar on the right is for the cells where the spark was silenced. The difference is just dramatic. Myelin production absolutely plummets. This is the final verdict. The spark isn't just for show. It's essential for the cell to do its main job.
So, let's put all the pieces of our investigation together. The verdict is in. This spark is absolutely vital. This electrical activity isn't some weird quirk. It's a required signal for these cells to mature, to grow their complex branches, and ultimately, to make the myelin our brains need to function. This discovery completely rewrites the rulebook. These aren't just passive helpers. They are active partners in a constant electrical conversation with neurons.
And that leaves us with this one last, really fascinating thought. We just found out that cells we'd written off as just support staff are actually sparking with electricity and actively listening to neurons. And it just forces you to wonder, what other incredible secrets are the brain's quietest cells still hiding from us?